Imagine standing at the edge of the ocean at night. The water is black, the horizon is gone, and the line between sea and sky has dissolved into something seamless and indifferent. Now imagine there is no shore beneath your feet. No sand, no rock, no ground at all. There is nothing behind you either. No city, no field, no forest fading into the distance. There is only water. Water in every direction. Water to the bottom of everything. Water for billions of miles in all directions, closing over you, beneath you, around you, with no interruption, no exception, no edge. You are not standing at the edge of the ocean. You are inside it. You are inside the only thing there is. That is what this planet looked like for most of its existence. Not most in the way we casually say most of my life. Most in a way that makes every human civilization, every mountain range, every desert and river valley and reef system look like a footnote written in the final paragraph of a story that took an incomprehensibly long time to tell. The Earth is approximately 4.5 billion years old. For roughly the first 4 billion years of that existence, give or take hundreds of millions depending on how you count, there was no stable, continuous land. There were no continents in any form we would recognize. There were no beaches, no cliffs, no plains, no valleys. There was, in the beginning, almost nothing but ocean. And yet life was there. This is the thing that should stop you. Not the absence of land. That is simply geology. The slow mechanical business of plate tectonics and crustal formation. The kind of fact you accept and move on from. What should stop you is the other part. The life, the fact that in that cold, pressurized, chemically alien, sunless, ancient water, something happened that has never fully been explained. Something that has never been replicated. Something that, once it began, never stopped. Life started in that ocean, found its footing diversified, built entire ecosystems, transformed the chemistry of the planet, and did all of this without a single organism ever setting foot on dry land. Because there was no dry land to set foot on. We spend a great deal of time thinking about the history of life on land. The dinosaurs, the mammals, the first lungfish dragging itself up a riverbank into the Devonian air. We think about evolution as a story that begins in the sea and arrives, triumphantly, at complexity, at consciousness, at us. But the version we tell ourselves leaves out almost everything. The story of life on land is a very short epilogue to a story that played out over billions of years in conditions we can barely imagine, written by organisms we barely have names for, in an ocean that no longer exists. So let us go back. All the way back. Before the fish, before the algae, before even the simplest cell we might recognize as ours. Back to when the planet was so young it was still cooling. Still being bombarded by the last debris of solar system formation, still figuring out what it wanted to be. Back to a time so distant that the light from those events, if light could carry information that far without distortion, would show us a planet that looks nothing like home. There is a moment, somewhere in the deep past, where everything that will ever live has not yet begun. And then, in the water, something shifts. That shift is what we are here to trace. The question of how life began is not a small one. It is arguably the largest question in all of biology, and it remains genuinely, stubbornly open. Not in the sense that scientists have no ideas. They have dozens of ideas, rigorously tested, hotly contested, with passionate advocates and devastating critics on every side. Open in the sense that no single explanation has closed the debate. No experiment has perfectly replicated the conditions. No discovery has made the others obviously wrong. The origin of life is still, in the most honest scientific sense, a mystery. And a mystery of this magnitude deserves to be approached not with the brisk confidence of a textbook summary, but with the kind of slow, careful attention you'd give to something genuinely precious. Something you might only get to see once. Have you ever noticed how strange it is that life is so common and yet started, as far as we know, exactly once? Every organism alive today, from the archaea living in boiling acid springs to the blue whale, from the oak tree to the tardigrade, from you to the bacterium currently living in your gut, all of it shares a common ancestor, one line, one beginning. The entire riot of life on Earth traces back to a single original event in a body of water that no longer exists on a planet that no longer looks the way it did. There is something almost impossible about this. And yet here we all are, undeniably connected, undeniably alive, descendants of whatever first crossed that threshold billions of years ago. We are children of the deep water. All of us, and most of us have forgotten. Let's begin at the beginning, the real one. The Earth formed approximately 4.54 billion years ago from the collision and accretion of gas, dust, and rocky debris that orbited a young star, the star we now call the Sun. The early Earth was not a hospitable place by any familiar standard. It was hot. Catastrophically, incomprehensibly hot. The energy released by constant bombardment from space, combined with the heat generated by radioactive decay within the planet's own forming interior, kept the surface largely molten for the first several hundred million years. Scientists call this period the Hadean Eon, named for Hades, the Greek underworld, which is fitting, though perhaps even that name undersells how extreme conditions were. The surface of the early Earth was an ocean of magma. Not metaphorically, literally. A planet-wide sea of liquid rock, glowing, churning, releasing gases that formed the earliest version of an atmosphere, mostly water vapor, carbon dioxide, methane, ammonia, sulfur dioxide. No free oxygen, no ozone layer, ultraviolet radiation from the young sun hammering down on everything, unfiltered. And yet water arrived. This is one of the foundational puzzles of early Earth science. Where did the water come from? For a long time, the dominant hypothesis was that most of Earth's water was delivered from space, carried by icy comets and water-rich asteroids that bombarded the planet during its first few 100 million years. The bombardment was so intense during the period called the Late Heavy Bombardment, roughly 4 to 3.8 billion years ago, that astronomers have a name for it, and it left a record on the pockmarked surface of the Moon which has no geological activity to erase those ancient scars. Water, on this view, came from the outer solar system, locked inside frozen bodies that fell from the sky. More recent research has complicated this picture. Isotopic analysis, essentially reading the chemical fingerprints in ancient minerals and comparing them to those of comets and asteroids, suggests that much of Earth's water may have been present from the very beginning, incorporated into the planet as it formed trapped in minerals and slowly released as the planet degassed. The debate continues. The answer may be both. The point is, by roughly 4.4 billion years ago, possibly earlier, liquid water was present on the surface of the Earth. We know this because of a mineral called zircon, a crystal so durable it persists through almost every geological process that would destroy other evidence. And the oldest known zircons found in the Jack Hills of Western Australia are dated to approximately 4.4 billion years ago. Their chemical composition suggests they formed in the presence of liquid water. The ocean was old before almost anything else we can point to. The first ocean was not the ocean we would recognize. It was probably much hotter, much more acidic, much more chemically active. The atmosphere above it bore no resemblance to ours. No oxygen, a toxic mix of greenhouse gases that kept the planet warm despite the fact that the young sun was considerably dimmer than it is today. The ocean interacted constantly with a still geologically active seafloor, with volcanic systems venting chemicals and heat from below. The water was rich with dissolved minerals, gases, metals. It was, in the language of chemistry, a reactive environment. And in reactive environments, things happen. The question is what things, and when. The first life, whatever it was, left no direct fossil record. Fossils require organisms with durable parts, shells, bones, mineralized structures, or conditions so exceptional that soft bodies become preserved in rock. The earliest organisms had no hard parts, and the rock record from that time is either destroyed, deformed, or simply missing. What we have instead are chemical traces, molecular fossils, ratios of carbon isotopes in ancient sedimentary rocks that suggest biological processes were operating. The oldest widely accepted evidence for life on Earth comes from rocks in the Pilbara region of Western Australia, dating to approximately 3.5 billion years ago. Stromatolites, layered structures built by microbial communities that trapped and bound sediment. Some researchers push the evidence even further back, to 3.7 or even 4.1 billion years ago, though these older claims remain contested. But consider what this means. The ocean was present by 4.4 billion years ago. The first clear evidence of life appears by 3.5 billion years ago, possibly much earlier. That is a window of roughly a billion years during which the transition from chemistry to life took place. A billion years, and yet we have no clear record of it. The most important event in the history of this planet, arguably the most important event in the history of anything we know about in the universe, happened in a window so ancient the rocks themselves have been recycled, subducted, metamorphosed, and largely erased. We are left with hypotheses, experiments, arguments, and a deep, persistent not knowing. That not knowing is not a failure of science. It is the honest acknowledgement of how far we are from the event, and there is something right about sitting with that honesty for a moment before rushing to fill the gap with answers. The water was there. Life appeared in the water. Between those two facts lies a billion years of chemistry that became something other than chemistry. Something that could grow, replicate, change, persist. Something that wanted, in whatever sense an entity without consciousness can want anything, to continue. We don't know exactly how that happened. But we have learned, in the last century of inquiry, a great deal about the conditions under which it could have happened. And those conditions are stranger and more beautiful and more violent than almost anything you've been told. Here is a useful way to think about what separates a living thing from everything else. It is not complexity alone. Crystals are complex. It is not the ability to reproduce. Fire reproduces, in a sense, spreading from source to source. It is not even the use of energy. The weather uses energy. What makes a living thing different is something more specific and more strange. A living thing is a system that uses energy to maintain itself against the tendency of everything else to fall apart. More than that, it carries information about how to keep doing that. And more than that again, it can copy that information imperfectly, introducing variation, creating the substrate upon which selection can act. Life, at its most stripped down, is chemistry that learned to remember itself. That description sounds clinical, and the actual business of how that happened is anything but. The question of the origin of life is one of the most contentious in all of science. Not because scientists disagree about whether it happened, the evidence is overwhelming that it did, but because the path from simple chemistry to the first self-replicating system is almost vanishingly difficult to trace. Every proposed solution runs into a version of the same problem. The thing you need in order to create the system requires the system to already exist. Take proteins, for example. Proteins are the workhorses of living cells. They act as enzymes, speeding up chemical reactions that would otherwise take millions of years to occur. They build structures, carry signals, regulate which genes are expressed. Proteins are, in many ways, what makes life metabolically possible. But proteins are built according to instructions encoded in DNA. And DNA, the information molecule, is copied and repaired by proteins. Which came first? You need proteins to read the DNA. You need DNA to make the proteins. This is the molecular equivalent of the chicken and egg problem, and it is not a casual paradox. It is a genuine logical barrier that stumped origin of life researchers for decades. The resolution, when it came, was not what anyone expected. In the 1980s, two researchers, Thomas Cech and Sidney Altman, working independently, discovered something that won them both the Nobel Prize. They discovered that RNA, the molecule that sits between DNA and proteins in the modern cell, can act as both an information carrier and a catalyst. RNA can fold into complex three-dimensional shapes, just like a protein, and those shapes can catalyze chemical reactions. RNA can also serve as a template for its own replication. It can, in other words, do both jobs at once. It can be both the chicken and the egg. This led to what became known as the RNA world hypothesis. The idea that the first self-replicating systems on Earth were not DNA-based but RNA-based. That before the complex division of labor we see in modern cells, DNA for storage, RNA for messaging, proteins for doing, there was a simpler, more unified system in which a single type of molecule carried information and acted on it. An RNA world, in other words. A world in which the first protocells were built around molecules that could both encode and execute. The RNA world hypothesis is, today, the most widely accepted framework for thinking about the origin of life. But it is not without problems. RNA is a chemically fragile molecule. It degrades quickly in water unless conditions are right. Getting it to replicate accurately without an existing biochemical machinery is extraordinarily difficult. And getting the building blocks of RNA, the nucleotides, to assemble spontaneously under plausible early Earth conditions requires a very specific sequence of chemical events. Researchers have made stunning progress on this problem in recent years. Work by John Sutherland and his colleagues at the MRC Laboratory of Molecular Biology in Cambridge has shown that certain nucleotides can be synthesized under conditions that may have existed on the early Earth, under ultraviolet light, in the presence of cyanide and phosphate with subtle but achievable chemistry. The gap is narrowing, but it has not closed. So where did this chemistry happen? What was the environment that first gave RNA, or whatever came before it, the conditions it needed to form and begin copying. Two answers have dominated the field, and they are as different from each other as any two places on Earth could be. The first is the warm little pond. Darwin himself speculated about it in a letter written in 1871, imagining life beginning in some warm little pond, with all sorts of ammonia and phosphoric salts, light, heat, electricity, and so on. The modern version of this idea focuses on tide pools, shallow coastal waters, or terrestrial hot springs. The argument is that in shallow environments, wet-dry cycles could concentrate organic molecules that would otherwise be too dilute to react. When water evaporates, the chemicals left behind come into closer contact. When water returns, the reactions can proceed. It is a kind of cyclical pumping mechanism, repeatedly concentrating and diluting, until something useful happens. Some researchers have argued that this accounts for many of the assembly problems, the difficulty of getting nucleotides to link together into long chains, for example, is reduced if you repeatedly concentrate them on mineral surfaces. The second answer is the deep sea vent. And this is where the story becomes genuinely extraordinary. In 1977, a research expedition diving in the submersible Alvin near the Galapagos Rift in the Pacific Ocean made a discovery that nobody had expected, and that overturned a foundational assumption about where life could exist. They found hydrothermal vents on the ocean floor. Not particularly unusual in themselves, scientists knew hydrothermal vents existed. What nobody expected was what was living around them. Clusters of life. Extraordinary, dense, teeming clusters of life. In water nearly 2,500 meters below the surface, in total darkness, far beyond the reach of sunlight. Giant tube worms 2 meters long, clams the size of dinner plates, shrimp, crabs, fish, whole communities of organisms thriving in conditions that were supposed to be uninhabitable. The question of how they survived without sunlight led to the discovery of chemosynthesis, the process by which certain bacteria use the chemical energy in hydrogen sulfide and other compounds emitted by the vents to fix carbon dioxide into organic matter. Just as plants use sunlight, these bacteria formed the base of entire ecosystems that ran on chemistry rather than light. The implications were enormous. Life did not need sunlight. Life could thrive in the deep ocean. Life could exist anywhere there was a chemical energy gradient, a difference in chemical potential that organisms could exploit. And this, in turn, reopened the question of where life began. The black smokers found in 1977 were not the most interesting vents for the origin of life. They were searingly hot, up to 400 degrees Celsius, acidic, rich in heavy metals. The chemistry was so intense that complex molecules would be destroyed almost as fast as they formed. But in 2000, a different kind of vent system was discovered in the mid-Atlantic, the Lost City hydrothermal field. Lost City is different from a black smoker in almost every way. It is not driven by magmatic heat, but by a chemical reaction called serpentinization, the reaction of seawater with ancient olivine-rich rocks, which produces heat, alkaline water, and large quantities of hydrogen gas. The temperature of Lost City vents is much lower, between 40 and 90 degrees Celsius. The water is alkaline, not acidic. The structures they build are not black plumes but white towers of carbonate minerals, some as tall as 60 meters, fragile-looking and magnificent. Mike Russell, a geologist and astrobiologist now at NASA's Jet Propulsion Laboratory, had been developing a theory about the origin of life at alkaline hydrothermal vents long before Lost City was discovered. And when it was discovered, it looked disturbingly like what he had predicted. His theory, developed with colleagues over decades, proposes that the first protocells did not need to synthesize RNA in a test tube. Instead, they used the natural proton gradients at alkaline vents, the difference in acidity between the alkaline vent fluids and the slightly acidic early ocean, as a source of energy. This gradient, Russell and his colleagues argue, is essentially the same kind of energy gradient that all living cells still use today. The mitochondria in your cells run on a proton gradient. The ATP synthase enzyme that makes the molecule that powers all your cellular activity is driven by protons flowing through a membrane down a concentration gradient. We still use the energy of the ancient vent. The idea is that the mineral pores within vent structures acted as the first protocell membranes, tiny compartments, nanometer-scale spaces within the rock where chemistry could be concentrated, where gradients could be exploited, where the first metabolic reactions might have occurred. Not in a pond on the surface, but in the sheltered, chemically driven labyrinth of a deep-sea vent, powered by the geological energy of a young, tectonically active planet. The debate between shallow, warm ponds and deep alkaline vents has not been resolved. Both environments have virtues. Both face serious objections. Some researchers are not convinced that either is the full answer and are exploring hybrid models or entirely different environments. What is agreed is this: whatever happened, happened in water, in the ocean, in conditions that no longer exist anywhere on Earth, on a planet that no longer looks the way it did. The fact that we are here, having this conversation, is the proof. But proof of outcome is not the same as understanding of mechanism, and the mechanism still eludes us. Have you ever sat with a problem so large that the not knowing itself becomes a kind of presence? The origin of life feels like that. It is the kind of question that you can hold in your mind for a moment, and then have to put down, not because it is too difficult to think about, but because it is too important to treat lightly. Something happened, in some body of water, some impossible number of years ago, and we are its consequence. Every cell in your body is the descendant of that event. Whatever that first molecule was, whatever crossed the line from chemistry to life, it never stopped. It became everything. That is not nothing. That is perhaps the most significant thing there is. Let's say life began. Let's say, for the sake of moving forward, that somewhere in the Hadean or early Archean ocean, in the pores of a mineral-rich vent or in the edge of a shallow tidal pool or in some environment we haven't imagined yet, the first self-replicating molecule formed. Let's say it copied itself. Imperfectly, but consistently. Let's say that variation arose, that some copies worked better than others. That selection began acting on the difference, and that from this humble, chemical beginning a lineage was established that has never since been broken. What did that first life look like? It looked like nothing we have a word for. The closest modern analogy is a prokaryote, a cell without a nucleus. But even that term implies a level of organization that may not have been present at the very start. The earliest living systems were almost certainly simpler than any cell we can observe today. Perhaps they were just membranes enclosing a small volume of water, with RNA molecules inside that could catalyze their own replication. Perhaps the membrane itself came first, lipid molecules spontaneously assembling into bilayers, which they do quite readily, because of the physics of how hydrophobic and hydrophilic ends of a fatty acid molecule interact with water. Perhaps the RNA came first and the membrane came later. Perhaps the first metabolic chemistry happened not inside a cell at all, but in the pores of a mineral, and the cell membrane was a later development that allowed mobility and independence from the vent. We don't know. What we do know is that within a few 100 million years of the planet cooling enough to support liquid water, which means roughly 4 to 3.5 billion years ago, and possibly earlier, life was present in the ocean. Not life as a marginal, tentative experiment at a single site. Life as a distributed, global phenomenon. The evidence for this comes from carbon isotope ratios preserved in ancient sedimentary rocks. Carbon-12 is preferentially incorporated into organic matter by biological processes, and ancient rocks from the early Archean show carbon-12 enrichment patterns consistent with life. The life was microscopic, unicellular, and metabolically diverse in ways that are not yet fully understood. The Archean eon spans from approximately 4 to 2.5 billion years ago. For that entire period, a span of time, 1.5 billion years long, longer than the time between the explosion of animal life in the Cambrian and the present day, the most complex organism on Earth was a single cell. Sometimes those cells lived in communities, sometimes in remarkable layered structures called stromatolites, but each individual organism was still just one cell, with no nucleus, no organelles, no compartmentalization of function beyond the membrane that enclosed it. This might seem like stasis. It was not. During the Archean, life was doing something far more important than getting bigger or more complex in any visible sense. It was diversifying metabolically. It was exploring the chemical space available to it, finding new ways to extract energy from an environment that presented enormous chemical challenges. And in doing so, it began to change the planet. The early ocean was chemically stratified in ways we don't fully understand. The atmosphere above it was almost entirely devoid of free oxygen. This is crucial, and we will return to it, which means the ocean was reducing, full of dissolved iron and other metals that would precipitate out as soon as oxygen appeared. The early life operated in this reducing world. The first metabolisms were probably based on chemical energy: hydrogen, hydrogen sulfide, iron, other inorganic compounds, rather than light. These chemolithotrophs, as they are called, life forms that eat rocks in the most literal chemical sense, were the earliest metabolic pioneers. And then, somewhere around 3.5 to 3.2 billion years ago, the dates remain uncertain, something new appeared. Something that would eventually change everything. A form of life that could use sunlight directly as an energy source. Not the kind of photosynthesis you learned about in school, not yet. The earliest photosynthesis was anoxygenic. It did not produce oxygen as a byproduct. Instead, these early photosynthesizers used sunlight to drive chemical reactions that extracted electrons from hydrogen sulfide or ferrous iron rather than from water. They were exploiting the energy of the sun, which was a revolution in itself, but they were not yet manufacturing the oxygen that would transform the planet. Think about what photosynthesis represents. When you strip it down to its essence, it is the ability to reach up through the water column, or out through the air if you're above the surface, and grab energy from a nuclear reaction happening 150 million kilometers away. It is one of the most audacious things any organism has ever done, coupling its metabolism to the output of a star, pulling light out of a vacuum and turning it into molecular food. The scale of this achievement, if you let it land for a moment, is staggering. A single cell, with no plans, no intelligence, no intention, evolved a set of molecular machinery that harvests starlight and uses it to stay alive. The stromatolites are the most visible legacy of these early photosynthesizers. The word comes from the Greek for layered rock, and that is exactly what they are. Layered, dome-shaped structures built by microbial mats, typically cyanobacteria and related organisms, that trap and bind sediment as they grow. The individual organisms grow upward toward the light. The mat grows, sediment falls, the mat grows over the sediment, more sediment falls layer by layer over thousands of years, building structures that can be meters high. Living stromatolites still exist in Shark Bay in Western Australia, in the Bahamas, in a few other places where conditions are unusual enough to prevent the grazing organisms that would otherwise consume them. But they are rare. When they were dominant, they covered vast stretches of the shallow Archean Ocean. The oldest stromatolites date to approximately 3.5 billion years ago. Some researchers claim even older examples, though the interpretation is contested. At that age, it can be difficult to distinguish a biologically constructed layered structure from one that formed purely through chemical processes. But by 3.5 billion years ago, there is strong evidence for organized, photosynthetic microbial communities building permanent structures in the shallow ocean. Already, life was leaving marks on the rocks that would persist for billions of years. And yet it was still just bacteria, still just single cells in a reducing, oxygen-free ocean on a planet with no continents, under a sky the color of orange haze, with no predators, no plants, no animals, no multicellular organisms of any kind. Just the hum of microbial metabolism, the slow construction of mineral mats, the patient, ancient business of life learning what it could do. There is something almost unbearable about this patience. We live in a time when change happens so fast that we struggle to keep up, when technology reshapes civilization within a generation, when entire species are driven to extinction in decades. Against that backdrop, the Archean Ocean asks us to slow down to a timescale so vast it becomes meaningless in human terms. For a billion years, nothing new appeared at the organismal level. No innovation in body plan, no new type of cell, no new form. Just variation within the bacterial theme. Metabolic diversity flourishing in the dark. Life doing the unglamorous, essential, world-transforming work of simply persisting. But something was building in that persistence. Something was accumulating. In the sediments and the rocks, in the chemistry of the water, in the slow creep of biological products into a world that had not encountered them before. The ocean was being altered. The planet was being altered. And the organism responsible for the most dramatic alteration of all was, at this moment in the story, still hovering on the edge of evolution. The thing that would change everything had not yet appeared, but it was coming. The word catastrophe suggests sudden, violent transformation. An avalanche, a meteor impact, a volcanic eruption that darkens the sky for years. The event we are about to describe was catastrophic in its consequences, but it unfolded with the slowness of geology. A slow-motion catastrophe that played out over hundreds of millions of years, visible only in the rock record. Felt by organisms that had no capacity to feel anything except the most fundamental biological imperatives of growth, reproduction, and survival. It began with a bacterium. Cyanobacteria are not dramatic-looking organisms. Under a microscope, they look like small cylinders or spheres, sometimes arranged in chains or clusters, sometimes forming filaments, sometimes embedded in a mucilaginous matrix. They are not particularly large even by bacterial standards. They have existed in recognizable form for at least 2.7 billion years, and possibly considerably longer. And they are responsible for what may be the single most transformative event in the history of life on this planet: the invention of oxygenic photosynthesis. The previous chapter mentioned an oxygenic photosynthesis, the kind that doesn't produce oxygen, that steals electrons from hydrogen sulfide or iron rather than from water. Oxygenic photosynthesis is different, and the difference matters enormously. In oxygenic photosynthesis, the electron donor is water. The cell takes in water, uses the energy of sunlight to split the water molecule apart, uses the electrons for its metabolic chemistry, and releases the leftover oxygen as a waste product. Oxygen, the gas that seems so fundamental to life on Earth that it's hard to imagine the planet without it. But for the organisms living in the Archean Ocean, oxygen was not life. It was poison. This requires a moment of reflection, because we are so accustomed to thinking of oxygen as necessary that the idea of it as a toxin feels counterintuitive. But oxygen is chemically extremely reactive. It attacks biological molecules, breaks them down, combines with metals, oxidizes the building blocks of the biochemistry that all early life had evolved to function in. An organism that evolved in a low oxygen or oxygen-free environment, which was every organism on Earth for the first couple of billion years, is not prepared for suddenly being bathed in a highly reactive oxidant. To those organisms, oxygen was something close to bleach. The cyanobacteria began producing oxygen at some point. The date is debated, with estimates ranging from 3.0 to 2.7 billion years ago, but the atmosphere and ocean did not immediately fill with it. For a long time, perhaps hundreds of millions of years, the oxygen produced by photosynthesis was absorbed almost entirely by reduced materials in the ocean and in the crust. The ocean floor at the time was rich in dissolved ferrous iron, iron in its reduced form, which reacts readily with oxygen. When the cyanobacteria produced oxygen, it oxidized the iron, which precipitated out of solution as iron oxide and settled to the seafloor. The result is visible today in what geologists call banded iron formations, layered sequences of iron oxide rock found on every continent, some of them hundreds of meters thick, formed between roughly 3.5 and 1.8 billion years ago. They are the oxidized iron of the ancient ocean. They are, in a real sense, the rusted remains of a world that cyanobacteria spent hundreds of millions of years chemically transforming. But iron is not infinite. Eventually, and this eventually spans hundreds of millions of years, the ocean ran out of dissolved iron to absorb the oxygen. And once the iron was gone, the oxygen had nowhere to go but up, into the atmosphere. The Great Oxidation Event, as it is now called, occurred approximately 2.4 billion years ago. The evidence comes from the rock record in several converging ways. The disappearance of minerals like pyrite and uraninite from river sediments, minerals that can only survive in the absence of atmospheric oxygen. The shift in sulfur isotope ratios that record the presence of an ozone layer, and the change in the oxidation state of iron in ancient soils. Something shifted, approximately 2.4 billion years ago, and the atmosphere of the Earth was never the same again. Oxygen accumulated. The ozone layer began to form. The planet was, from that moment, fundamentally altered. The consequences for life were enormous. For the anaerobic organisms that had dominated the planet until that point, the rise of atmospheric oxygen was potentially catastrophic. Obligate anaerobes, organisms that cannot tolerate oxygen, were driven into refuges. Deep in sediments, in the gut of other organisms in oxygen-depleted corners of the ocean and the soil, many lineages may have gone extinct entirely. This was, from one perspective, a mass extinction, the first mass extinction in the history of life, caused not by an asteroid or a volcanic eruption, but by the metabolic byproduct of a microorganism that was simply trying to eat sunlight. But the Great Oxidation Event also opened the door to something new. Oxygen is a powerful electron acceptor, which means it can serve as the basis for a much more energetically efficient form of metabolism than anything that had come before. Aerobic respiration, the kind that you use, the kind that runs every animal cell on Earth, is roughly 15 to 18 times more energetically efficient than the anaerobic alternatives. The chemical energy available to an organism that can breathe oxygen is vastly greater than the energy available to one that cannot. The rise of oxygen, in other words, was a catastrophe for some and a gift for others. It was a reset, a door closing on one world and opening on another. There is something profound in this that extends well beyond biology. The thing that ended one era of life was precisely the metabolic waste product of life itself. The organisms living in the Archean Ocean did not intend to change the planet. They had no intentions at all. They were just doing what life does, finding energy, using it, producing byproducts, growing, reproducing, and in doing this blindly over hundreds of millions of years. They altered the chemistry of the atmosphere and ocean so completely that they made their own environment uninhabitable for many of their contemporaries, and set the stage for forms of life that could not have existed in the world they were destroying. Life does not merely inhabit environments. Life creates them. Life changes them. Life, at its most patient and persistent, is a geological force. We carry the legacy of this in our lungs. Every breath you take is built on the metabolic waste of ancient cyanobacteria. Every oxygen molecule entering your bloodstream right now was, at some point in the deep past, produced by an organism you will never see, in an ocean that no longer exists, on a planet you would not recognize. The air is ancient. The chemistry is borrowed from something older than anything you can imagine. It is perhaps worth pausing on that, just for a moment, before moving on. But there is more to tell. The Great Oxidation Event was not the only seismic change in the Archean and Proterozoic oceans. There was another event, roughly contemporaneous, that was in some ways even more consequential for the long-term history of life. Not a chemical change in the atmosphere, a change inside the cell itself. It is the story of a merger that should not have worked, and yet became the foundation of every complex life form that has ever existed. There is a moment in the history of life that is so improbable, so structurally strange, so unlike anything that had happened before or would happen again in quite the same way, that every time biologists look at it closely, they seem to see something new. It is the moment when one cell ate another cell and didn't digest it. Or possibly the other cell invaded the first one and didn't kill it. The relationship of host to guest is still unclear, still debated, and perhaps the question of who ate whom misses the point. What happened was a merger, a fusion, an event in which two profoundly different kinds of life became one life. And that one life became the ancestor of everything you have ever seen or touched or loved or eaten. Let's back up slightly. For most of the Archean eon and into the early Proterozoic, life on Earth consisted entirely of prokaryotes, cells without a nucleus, without membrane-bound organelles, without the intricate internal architecture that characterizes the cells of your body. Prokaryotes are not simple. Their biochemistry is extraordinarily sophisticated. Their genetic regulation is complex. Their ecological roles are critical and irreplaceable. But they lack the organizational elaboration that defines eukaryotes. They have no nuclear membrane. They have no mitochondria. They have no organized internal membrane system. They are, in comparison to a eukaryotic cell, architecturally minimal. Eukaryotes are different. The word means roughly true kernel, a reference to the nucleus, the membrane-enclosed compartment at the center of the cell that contains the DNA. But the nucleus is only part of what makes a eukaryote different. The other thing, the more important thing for the story we're telling, is the mitochondrion. Mitochondria are the energy factories of eukaryotic cells. Every biology student knows this. They carry out aerobic respiration, produce ATP, sustain the energy demands of complex cellular life. And they have a history that is both bizarre and, in retrospect, inevitable. Mitochondria were once bacteria. This idea was proposed and developed most forcefully by the American biologist Lynn Margulis in a 1967 paper that was rejected by over a dozen journals before finally being published. The resistance was understandable. The idea that a fundamental and universal feature of complex life was the product of ancient bacterial endosymbiosis, one cell swallowing another and keeping it alive inside, was, to put it mildly, unconventional. Evolutionary theory at the time was focused on gradual change within lineages, not wholesale mergers between them. Margulis was saying something that didn't fit neatly into any existing category. She was right. The evidence is overwhelming, and it comes from multiple directions. Mitochondria have their own circular DNA, just like bacteria. They have their own ribosomes, distinct from the ribosomes in the rest of the cell. They divide by binary fission, as bacteria do, not by the cell cycle mechanisms that divide the nucleus. They are bounded by two membranes, which is consistent with their origin as a gram-negative bacterium enveloped by the host cell's membrane. Phylogenetic analyses comparing the DNA sequences of mitochondria to those of living bacteria Identify the ancestors of mitochondria most likely within the group called alpha protobacteria. Mitochondria are not metaphorically bacterial. They are, in every meaningful biological sense, reduced and co-opted bacteria living inside your cells. The origin of mitochondria probably occurred around 1.5 to 2 billion years ago. The merger, whatever form it took, transformed the energetic capacity of the resulting cells so dramatically that it opened entirely new possibilities. With mitochondria, the host cell could do aerobic respiration. It could extract dramatically more energy from the same molecules. It had the metabolic headroom to afford larger genomes, more complex regulation, more elaborate cellular architecture. The cell that had mitochondria could become complicated in ways that the cell without them could not. And chloroplasts, the organelles in plant cells that carry out photosynthesis, have the same origin story, just a second telling of it. The ancestors of chloroplasts were cyanobacteria, a later endosymbiotic event, in which an early eukaryote engulfed a cyanobacterium and kept it too, gave the eukaryotic lineage that would become algae and plants the ability to photosynthesize. The entire photosynthetic apparatus of every plant on Earth, every leaf, every blade of grass, every algae-covered rock in a stream, is built on enslaved cyanobacteria. We are, in this sense, composite organisms, not in a metaphorical way, in a literal, historical, genomic way. The cells of your body are the outcome of multiple ancient mergers between organisms that were, at the time of the merger, separate living entities with separate evolutionary histories. You are a colony. You are a negotiated truce between lineages that once competed or, at minimum, inhabited different parts of the world without any connection at all. The cells inside you still carry echoes of that history. The mitochondrial genome, the bacterial-style ribosomes, the double membrane that marks them as guests from another kingdom. Margulis called this endosymbiosis, and she spent her career extending the idea far beyond mitochondria and chloroplasts, arguing that major evolutionary transitions are often driven by symbiosis and merger rather than gradual change within a single lineage. This view was initially controversial. It is now central to our understanding of eukaryotic evolution. The merger that made us is no longer a fringe idea held by an eccentric biologist. It is the established, well-supported foundational fact of complex cellular life. But what does it mean? What does it mean for how we think about individuality, about boundaries between organisms, about what a self even is? A cell that contains an ancestor of a separate bacterium and that cannot live without it, and that has integrated its metabolism with it so completely over 1 billion years that removing it is impossible. Is that one organism or two? The question is not pedantic. It points to something real about how life operates. Life does not keep clean boundaries. It merges. It cooperates. It invades and is invaded. The boundaries between organisms are far more porous than any diagram in a biology textbook would lead you to believe. And this porosity is not incidental. It is possibly the engine of major innovation. Every complex organism on Earth is a chimera built from ancient partnerships. The trees, the fish, the fungi, the insects, the mammals, all of them descended from that original merger, that moment in the Proterozoic ocean when one cell kept another alive inside itself rather than digesting it. We do not know exactly when or where or how many times this happened. We know it happened, and we know that without it, we would not exist. There is a kind of debt here that cannot be repaid. To the ancient bacterium that became the mitochondrion, to the ancient cyanobacterium that became the chloroplast, to whatever unnamed, unknown host cell opened itself to these borders and was changed by the encounter. They are gone now, those specific organisms. They exist only as the ghosts inside our cells. But they made us possible. They are, in the most intimate biological sense, part of us. That seems like something worth knowing. 2 billion years ago, 1.5 billion years ago, 1 billion years ago. These are numbers we say quickly and register almost not at all. 1 billion years is not comprehensible on human terms. The entire written history of humanity is something like 5,000 or 6,000 years old. The agricultural revolution was 10,000 years ago. The extinction of the dinosaurs was 66 million years ago, and yet life in the ocean, during what geologists call the Proterozoic eon, which spans from 2.5 billion years ago to 541 million years ago, operated on timescales that make all of these markers look like they happened this morning. The Proterozoic is sometimes called the boring billion, a name applied specifically to a middle period roughly 1.8 to 0.8 billion years ago, when the fossil record and geochemical proxies suggest that ocean chemistry was relatively stable. Atmospheric oxygen was still moderate and possibly declining, and there appears to have been a relative plateau in evolutionary innovation at the organismal level. Eukaryotes were present. Simple multicellular forms may have existed, but the explosion of complexity that defines the Cambrian, the sudden appearance in the fossil record of animals with skeletons and shells and complex body plans, had not yet occurred, and the intermediate steps remained frustratingly unclear. The name boring billion is unfair, and it is worth dwelling on why. The ocean during this period was not boring. It was chemically complex, biologically active, and undergoing changes at a pace that is simply invisible on human timescales. The deep oceans during much of the Proterozoic are thought to have been anoxic, and in some models, rich in hydrogen sulfide, what scientists call oceanic conditions. The shallow coastal oceans had some oxygen. The boundary between these zones moved up and down over time, responding to changes in photosynthetic productivity, in nutrient input from weathering of the early sparse continental surfaces, in the biology of the organisms living in and around the water. This was not a static, boring system. It was a dynamic, chemically stratified ocean with complex biological communities operating within it. And the organisms themselves were not standing still. The Proterozoic is when eukaryotes truly diversified. Simple algae, single-celled photosynthetic eukaryotes, appear in the fossil record of Proterozoic rocks, acritarchs, enigmatic microfossils that may represent the resting stages or cysts of various eukaryotic algae, become abundant and diverse. Red algae, one of the oldest lineages of multicellular algae, have fossil representatives that date to approximately 1.2 billion years ago in the rocks of the Hunting Formation in Arctic Canada, exquisitely preserved, showing a level of cellular complexity and organization that suggests multicellularity was already well established. There were organisms with nuclei, with organelles, with the molecular machinery for sexual reproduction. There were organisms beginning to explore the possibilities of cellular cooperation. Sexual reproduction deserves a moment of attention here, because it is easy to take for granted and deeply strange when you don't. Bacteria reproduce asexually. One cell divides into two, producing genetic copies. Genetic variation in bacteria comes from mutation and from a process called horizontal gene transfer, in which organisms pass genes directly to one another, not just to their descendants. But sex, as eukaryotes practice it, the fusion of two cells, the shuffling of genetic material from two distinct lineages into a new combination, is a completely different approach to generating variation. It is more expensive, metabolically. It requires finding a partner. It dilutes the representation of any individual's genes in the next generation by half. Given these costs, it took evolutionary biologists a long time to explain why sex persisted at all, and the explanation that the rapid generation of new genetic combinations helps organisms outrun pathogens and parasites in an evolutionary arms race is still not universally accepted. But what is clear is that the invention of sex, somewhere in the Proterozoic ocean, was a fundamental change in how evolution proceeded. It dramatically increased the rate at which new genetic combinations could be explored. It set the stage for the diversification that followed. The other major development of the Proterozoic that deserves attention is the emergence of true multicellularity. Not just the colonial behavior seen in some bacteria, where cells aggregate but remain metabolically independent. Not just the loose associations of cells in early eukaryotic algae. True multicellularity, in which cells differentiate, become specialized for different functions, and in which the death of an individual cell is no longer the death of the organism. This transition happened independently many times, in the lineage leading to plants, in the lineage leading to fungi, in the lineage leading to animals, possibly in lineages that went extinct without leaving modern descendants. It is not a single evolutionary event, but a recurring theme, an innovation that life kept rediscovering in different ways. The key insight about multicellularity is what it requires at the cellular level. Individual cells in a multicellular organism give up the freedom to reproduce on their own terms. A liver cell does not try to become a separate organism. It performs its function, it cooperates with its neighbors, and if it fails to receive the right signals, it dies, a process called apoptosis, programmed cell death. The entire body depends on cells agreeing, in a sense, to subordinate their individual interests to the needs of the whole. This is not cooperation in any conscious sense. It is the outcome of selection acting on genes that are present in every cell in the organism's body. But the logic of it is strange. Selection acting at the level of the organism can favor traits that are individually costly if they benefit the whole. The liver cell agrees to die because the organism that contains it survives better when liver cells die on schedule. Multicellularity also opened the door to size. A single cell is constrained in its dimensions by the physics of diffusion. Nutrients and gases can only travel so fast across a membrane and through the cytoplasm. A multicellular organism can be much larger because different cells can specialize in acquiring different resources and transporting them efficiently throughout the body. Size, in turn, opens ecological roles unavailable to the single cell. The ability to engulf large prey, to photosynthesize with a large surface area, to build structures that persist through time and provide habitat for other organisms. Size arrived slowly, but it arrived. The Ediacaran period, which immediately precedes the Cambrian, roughly 635 to 541 million years ago, shows the first clear evidence of macroscopic, complex multicellular life. The Ediacaran biota, named for the hills in South Australia, where many of the best fossils were found, are deeply strange organisms. They are soft-bodied. They leave no descendants we can clearly identify, and their relationship to modern animal groups remains a subject of genuine debate. Some appear to have been quilted, fractal structures, organized in ways that don't map onto any living body plan. Some were sessile, fixed to the seafloor. Some may have been photosymbiotic, carrying photosynthetic cells inside their bodies as some modern corals do. They are genuinely alien, these organisms, and yet they are the first chapter of the story that leads to animals. Have you ever felt like you were looking at something that you couldn't quite place? Something that doesn't fit into any existing category, that demands a new one? The Ediacaran biota produced that feeling in paleontologists. They lived in a world still almost entirely marine, still without any substantial terrestrial ecosystem. They lived in a shallow sea somewhere that is now South Australia, or what is now Newfoundland, or what is now Namibia or Scandinavia or Siberia. Ediacaran fossils have been found on every continent. They were strange. They were large by the standards of everything that had come before, and then at the boundary of the Cambrian, they were mostly gone. We do not fully understand why. The relationship between the end of the Ediacaran and the explosion of animal diversity in the Cambrian is one of the persistent puzzles of paleontology. Were the Ediacaran organisms wiped out by the newly evolving Cambrian animals, which could move and eat and bite in ways that the Ediacaran could not defend against? Did they go extinct for environmental reasons? Changes in ocean chemistry? Changes in oxygen availability? Did some of them give rise to Cambrian groups, and we simply can't see the connection clearly? The debate continues. The silence of the fossils is frustrating and absolute. But here, in the Proterozoic, before the Ediacaran even begins, something else is building in the ocean. Something quiet but transformative. The chemistry of the ocean is being gradually tuned by the organisms living in it. Carbon, oxygen, nitrogen, sulfur, all of them are cycling through living systems at rates that are subtly reshaping the environment. The ocean is not passive. The ocean is a living system, reacting to the organisms it contains, changed by them, channeling them in new directions. This dance between life and environment, between organism and the world, the organism simultaneously inhabits and creates, is one of the deep themes of the story we are telling. Life and Earth have been in conversation for 4 billion years. The ocean is the room in which that conversation takes place. And that room, slowly, over the Proterozoic and into the Ediacaran, was being prepared for something new. The name is dramatic and the event deserves it. Sometime around 541 million years ago, in the shallow seas of a planet that was beginning, in some places, to show hints of continental emergence, something happened that still lacks a fully satisfactory explanation. In the space of what geologists call an instant, which means, in this context, something like 20 million years, animal life diversified explosively. The number of distinct body plans represented in the fossil record went from a handful of soft-bodied Ediacaran forms to dozens of fundamentally different architectures. Animals with shells, with mineralized skeletons, with compound eyes, with grasping appendages, with bilateral symmetry and defined heads and tails and sides, with everything, in other words, that we associate with the basic organization of animal bodies, appeared in the rocks of the Cambrian in what looks from a geological perspective like an overnight revolution. The Burgess Shale, discovered in British Columbia in 1909 by Charles Walcott, is one of the great fossil treasures of the world. It preserves, with extraordinary fidelity, the soft-bodied organisms of a Cambrian seafloor community roughly 508 million years old. There are arthropods with elaborate spines and multiple eyes. There are primitive chordates, animals with a notochord, a stiffening rod that in our lineage became the backbone. There are organisms so bizarre that Stephen Jay Gould, who wrote about them in his 1989 book, described them as demonstrating that the history of life was not a progressive march toward complexity, but a wilder, more contingent, more improbable story. Organisms like hallucigenia, which paleontologists initially reconstructed upside down because nothing about it indicated which way was up. Organisms like opabenia, with 5 eyes and a flexible proboscis, which had the audience at a paleontology conference in 1972, laughing when it was first described, not from disbelief, but from the sheer improbability of the thing. Anomalocaris, the apex predator of the Cambrian, a meter-long swimming animal with compound eyes and grasping appendages, so unlike any modern arthropod that its parts were initially described as 3 separate organisms before someone realized they went together. What caused the Cambrian explosion? The question has been debated for over a century and remains genuinely open. Several factors are likely involved. The rise of atmospheric and ocean oxygen to levels that could support the metabolic demands of active, moving animals is one. The evolution of predation, the moment when one organism began actively hunting and eating other multicellular organisms, may have triggered an arms race that drove rapid diversification of defense structures and the skeletal elements and complex sensory systems needed to escape predators. Changes in ocean chemistry, particularly in calcium availability, may have made the construction of hard shells suddenly more energetically accessible. Ecological interactions, the diversification of one group creating new niches for others, may have fed on itself in a self-amplifying cascade. Whatever the cause or combination of causes, the result was the world we know. Not immediately, the Cambrian is still an ocean world, still entirely aquatic in its complex ecosystems. But the body plans, the fundamental architectures of animal life, are established in the Cambrian. The lineages that would eventually give rise to fish, to insects, to mollusks, to echinoderms, to every major group of animals alive today, they trace their origins to this period. We are Cambrian organisms at our deepest architectural level. The segmentation of your body, the bilateral symmetry, the placement of your eyes in your head, the basic layout of your nervous system. All of it was sketched out in ancestral form in the shallow Cambrian seas. The Cambrian seafloor was a complex, 3-dimensionally organized ecosystem. There were filter feeders and detritivores in the sediment, active predators swimming in the water column, sessile suspension feeders on the hard substrate. There were burrowing organisms, and the Cambrian is when the record of bioturbation, the physical disruption of sediments by burrowing animals, dramatically increases. Before the Cambrian, the seafloor was a laminated, relatively undisturbed environment. The microbial mats of the Proterozoic preserved in the sediment layers. After the Cambrian, the seafloor was churned, mixed, disrupted by organisms that burrowed through it looking for food or shelter. Even the sedimentary environment was permanently changed by animal life. The eyes, in particular, deserve attention. The Cambrian was when eyes appeared real, functional, image-forming eyes. The compound eyes of trilobites, some of the most complex visual organs known in the fossil record, evolved in the early Cambrian. Anomalocaris had large, stalked eyes. The first hints of the vertebrate eye lineage appear in early chordates of this period. Eyes are not just sensory organs. They are, in evolutionary terms, a transformation of ecological possibility. An animal that can see can hunt more effectively, can escape predators more effectively, can navigate a 3-dimensional environment. The appearance of vision changes the rules of the ecological game. Some evolutionary biologists, most notably Andrew Parker, have proposed that the evolution of eyes was itself the trigger for the Cambrian explosion, that once one predatory lineage could see its prey, an arms race began that drove the rapid evolution of defensive armor evasion behaviors and sensory countermeasures across many lineages simultaneously. This is the light switch hypothesis, and while it remains controversial, it captures something real about the transformative power of a single sensory innovation. But let's not get lost in the Cambrian. As tempting as it is, the Cambrian is, in a sense, the beginning of familiar history, the beginning of the story of animals. The story of the ocean before the Cambrian is the deeper story, the one we have been tracing, the one that runs from the first chemistry to the first cell, to the endosymbiotic merger, to the first multicellular organism, to the first macroscopic life. The Cambrian is the climax, but climaxes require what came before them to be fully understood. And what came before the Cambrian is still poorly understood, not for lack of trying. The Proterozoic fossil record is fragmentary, deformed, often chemically altered beyond recognition. The organisms of the deep past were overwhelmingly soft-bodied, and soft bodies rarely preserve. We see them when we see them at all in the form of chemical traces, isotopic signatures, occasional remarkable sites of exceptional preservation like the Dushantuo Formation in China, which has produced extraordinary microscopic fossils of AETERNA age, showing what appear to be the earliest stages of animal embryonic development. Cells dividing blastula-like clusters frozen in rock nearly 600 million years old. There is something almost unbearably tender about those embryos. Cells caught in the act of becoming something, preserved in rock for 600 million years, waiting for the one species that would eventually evolve the capacity to find them, extract them, examine them, and understand what they were. Life leaving a record of its own becoming. The ocean keeping archives. We should pause here and address something that has been present in the background of this entire story, but that deserves direct, careful attention. All of this life, the archean bacteria, the Proterozoic eukaryotes, the AETERNA giants, the Cambrian Explosion, played out in an ocean on a planet that looked almost nothing like the Earth we know, not just in terms of chemistry or atmosphere, in terms of its most basic physical geography. There were no continents, or rather, more precisely, there were no continents in any form that would be recognizable to us. The Earth has had some form of continental crust since early in its history. The Jack Hill zircons that told us about the ancient ocean came from ancient continental rocks, but the distribution size and stability of that crust bore no resemblance to the 7 large landmasses we know today. The early continents were small, scattered, geologically unstable, and repeatedly subducted, amalgamated, rifted apart, and reassembled. The process of plate tectonics, the engine that drives continental movement, that builds mountains, that opens ocean basins and closes them again, was operating from at least the Archean onward, though possibly in a form somewhat different from the modern style. Understanding the geography of the early Earth is extraordinarily difficult because the geological evidence is so fragmentary. Most ancient oceanic crust has been destroyed by subduction, pushed back into the mantle and recycled. What survives is the older, lighter continental crust, but even that has been deformed and metamorphosed and eroded beyond easy interpretation. We piece together the ancient geography from the rock record, from the paleomagnetic signatures in ancient rocks that tell us their latitude when they formed, from the sequences of sedimentary rocks that tell us about ancient seas and shorelines, from the patterns of ancient mountain belts and rift basins that tell us about collisions and separations. What we can say with confidence is that the planet during the Archean and much of the Proterozoic had much less continental surface area above sea level than it does today. Some models suggest that most or all of the continental crust was submerged or barely emergent during the early Archean, that the planet was for its first 1 billion or 2 billion years, essentially a water world, with only scattered volcanic islands or shallow banks breaking the ocean surface if anything did at all. The implications of this for the story of life are profound. Life was not simply confined to the ocean because organisms hadn't yet learned to live on land. Life was confined to the ocean because land in any substantial sense did not exist. The terrestrial biosphere, the entirety of life on land, which today constitutes an enormous fraction of global biodiversity and biomass, had nowhere to go. The land had not been prepared. Continents grew through time. The Archean craton nuclei, the ancient stable cores of continents, formed and stabilized during the Archean, and by 2.5 billion years ago, substantial portions of continental crust existed. But these were islands in a global ocean, small by later standards. The formation of large continental landmasses and their collision and assembly into supercontinents is a Proterozoic story. Rodinia, a supercontinent that may have formed around 1.1 billion years ago and rifted apart again around 750 to 700 million years ago, is the first supercontinent for which we have reasonable evidence. Before Rodinia, the configuration of whatever continental fragments existed is deeply uncertain. The global ocean of the Archean and early Proterozoic was almost certainly more connected than the modern ocean, with less continental area to break it into basins, with no deep mountain ranges on land to influence atmospheric circulation, with no plants on land to affect the water cycle. The ocean operated differently. Circulation patterns were different. The distribution of heat from the equator to the poles was different. The mixing of deep and shallow water, which today is driven partly by differences in temperature and salinity set up by continental geography, would have operated under different constraints. We are still building the models to understand this ancient ocean. The constraints are few, the degrees of freedom many. What we do know is that the life in this ocean evolved without any terrestrial reference point, without any evolutionary pressure to adapt to air, to desiccation, to ultraviolet radiation from a sky without an ozone layer. The earliest organisms were not planning a move to the land. They were not working their way toward it. They were adapting to the ocean that existed, optimizing for the conditions they found themselves in, generation by generation, over timescales that dwarf everything in human experience. The first hint of terrestrial life appears in the form of cryptic biological crusts on exposed rock surfaces, communities of cyanobacteria, and possibly early fungi that colonized bare rock wherever it was exposed to the air. These communities likely existed by the late Proterozoic, perhaps earlier. They were thin, fragile, dependent on intermittent moisture, far removed from the rich terrestrial ecosystems that would eventually develop. But they were there, and in the patient way that life has always operated, they were beginning the work that would eventually make the land habitable. But that is a story for another time, and a later Earth. For now, we are still in the ocean, still in the water, still in the world before the world we know. The continental shelves, the shallow, sunlit margins of whatever land existed, were always the most productive parts of the ancient ocean. Even in the Proterozoic, nutrient runoff from exposed rock, combined with the penetration of sunlight into shallow water, created environments richer in photosynthetic life than the open ocean. These shelves are where the stromatolites built their great reefs. These are where the Ediacaran organisms lived their strange, slow lives. These are where, in the Cambrian, the first explosion of animal diversity played out. The deep ocean, by contrast, was ancient, cold, pressurized, and largely disconnected from the light-driven productivity at the surface. It had its own ecosystems built around chemosynthetic bacteria, around the slow rain of organic material from above. But these were sparse by the standards of the shelves. The deep ocean was the closest thing to a stable, unchanging environment on a planet that was otherwise constantly transforming. And it is there in the oldest and most conservative of all Earth's environments that some of the earliest lineages of life may have persisted longest, sheltered from the changes above by the weight of the water itself. There is a metaphor here that seems almost too apt to be accidental. The deep ocean as refuge, the deep water as the place where ancient things survive, far below the disruption of the surface. We use that metaphor in human terms all the time. The deep as the place where secrets are kept, where the past is preserved, where the weight of the accumulated time bears down. And in the actual ocean, this is literally true. Some of the organisms living in the deepest reaches of the modern ocean belong to lineages so ancient that their morphology has changed little in hundreds of millions of years. The coelacanth, discovered living off the coast of South Africa in 1938, having been thought extinct for 65 million years, is one famous example. The ocean floor is a museum. The ocean keeps its oldest things in its deepest drawers. There is a chapter in the history of the ocean that is rarely taught in school, partly because it involves a kind of violence so large and so slow that the human mind struggles to hold it as something that actually happened, and partly because the evidence for it is recent and incomplete and still being argued over. But it is, in the opinion of many paleontologists and geochemists, among the most consequential events in the history of life on this planet. It is called Snowball Earth. Beginning perhaps 720 million years ago and possibly again around 635 million years ago, the Earth may have entered an extreme glaciation event, or two events, perhaps three, in which ice sheets advanced from the poles toward the equator and covered most or all of the planet's surface. If the most extreme version of this hypothesis is correct, the entire ocean was frozen over, covered by a layer of ice hundreds of meters thick, with only volcanic vents in the deep ocean providing any liquid water. If a somewhat less extreme version is correct, the so-called slushball Earth, some open water persisted near the equator, maintaining small refuges of liquid ocean in the tropics. The evidence for extreme glaciation comes from several sources. Glacial deposits, sedimentary rocks that record the physical movement of ice across the landscape, have been found in rocks of this age from multiple continents, including deposits that at the time of their formation were located at low latitudes near the equator, where ice should not have been able to form under any moderate climate scenario. Distinctive mineral signatures, including what are called cap carbonates, unusual layers of carbonate rock immediately overlying the glacial deposits, suggesting a dramatic and rapid return to warm conditions, also support the snowball hypothesis. The global distribution of these signatures, found on nearly every continent and rocks of the right age, suggests a truly global event. If the ocean was frozen, what happened to the life inside it? The honest answer is that we are still working this out. A complete global ocean freeze would have been catastrophic for photosynthetic life, since ice blocks sunlight. Without photosynthesis, the base of the marine food web collapses. Populations of algae and cyanobacteria would have crashed. Many lineages may have gone extinct entirely. But life did survive. The presence of complex, diverse organisms in the AETERNA, immediately following the snowball events, is proof that the catastrophe was not total. How did life survive? Possible refuges include the hydrothermal vents, where liquid water and chemical energy persisted regardless of surface conditions. Thin meltwater pools under the ice, where enough sunlight penetrated to support minimal photosynthesis. The deep ocean, where temperature is more stable and the effects of surface freezing are felt last. And possibly pockets of bare rock exposed in the high-altitude volcanic regions, where enough geothermal heat prevented complete freezing. The snowball events may have ended when carbon dioxide, which could no longer be absorbed by oceans or weathered by rocks, because both were covered in ice, accumulated in the atmosphere to extreme levels from continued volcanic outgassing. Eventually, the greenhouse effect of the accumulated CO2 would have tipped the climate back toward warmth. The ice would have melted rapidly, and the ocean would have emerged into a warmer than normal period. The cap carbonates record this dramatic transition, the sudden return to chemical weathering and carbonate deposition after the cold. The aftermath of snowball Earth may have had something to do with the AETERNA and Cambrian explosions of complex life. The idea proposed by various researchers in different forms is that the extreme bottleneck of the glaciation events drove rapid evolutionary change in the survivors, that the populations emerging from snowball refuges were genetically changed, that the new environmental conditions following deglaciation were dramatically different from the pre-glacial world, and that this combination of new genetic diversity and new environments triggered a burst of diversification. The timescale fits. The end of the last snowball event around 635 million years ago precedes the AETERNA biota by only a few tens of millions of years, and the Cambrian Explosion follows roughly 100 million years later. Whether the connection is causal, correlational, or coincidental remains debated. Origin stories for major evolutionary events always attract multiple competing hypotheses. But the idea that near-death experiences are sometimes followed by remarkable creativity is at least suggestive. That pressure, the kind that comes from being pushed to the edge of what is survivable, sometimes produces novelty. That the organisms that came through the bottleneck did so with a biological heritage shaped by the severity of what they had endured. This is not, of course, a lesson about resilience in the self-help sense. These organisms were not exercising will or choosing to adapt. They were the survivors of a selection process so brutal that most of their contemporaries did not survive it. The resilience was not theirs. It was evolution's. The lineages that happened to have the genetic variation that allowed some individuals to persist in some refuges in some minimal way happened to give rise to the organisms that came after. It was blind and mechanical and statistical, but it was also extraordinary. Something in the ancient ocean, in whatever water remained liquid under the ice, held on. Something did not go out, and from that small, cold, dark, pressurized persistence, the entire subsequent history of complex life descended. The ocean kept secrets and it kept them cold, and from those secrets it built the world. We've been talking about the deep ocean as a place where things were preserved, where ancient lineages sheltered. But the deep ocean was also, from early in its history, a place where life had evolved specific and remarkable adaptations to conditions that have no parallel anywhere else on Earth. And the story of those adaptations is a story that has only been properly told in the last 50 years, as technology finally gave us the ability to go there. The deep ocean begins, by convention, at 200 meters below the surface, the limit to which significant sunlight penetrates, the boundary of the photic zone, the depth below which photosynthesis is no longer possible. Below 200 meters, the ocean becomes darker, colder, and more pressurized with every additional meter of depth. At 1,000 meters, you are in the midnight zone, complete darkness, water temperatures typically between 2 and 4 degrees Celsius, pressure of 100 atmospheres. At the bottom of the Mariana Trench, the deepest known point in the ocean at just under 11,000 meters, the pressure is approximately 1,000 times the atmospheric pressure at the surface. The temperature is close to freezing. No sunlight has ever reached there. It is, in every measurable environmental variable, a harsh place to be alive. And yet life is there. Life is abundant there. Not abundant in the way of a coral reef or a kelp forest, where the sunlight drives such high rates of primary production that every surface is covered with organisms, and the water is thick with plankton, and the whole system vibrates with biological activity. The deep ocean is much more sparse than that, much more controlled in its energy budget. But it is far from empty. The organisms of the deep ocean have evolved over geological time to exploit the resources available in their extreme environment. Those resources are predominantly two: the slow rain of organic particles from the surface, marine snow as it is called, the accumulated dead cells fecal pellets, and organic aggregates that drift slowly downward from the productive surface layers, and the chemical energy released by hydrothermal vents and cold seeps along the seafloor. These two energy sources support very different communities, and both have ancient roots. The organisms that feed on marine snow, the deep-sea detritivores, the filter feeders, the scavengers, are part of a food web that connects the deep ocean to the surface in ways that are still being mapped. The biological pump, as oceanographers call it, is the process by which photosynthetic production at the surface is transferred to the deep. Carbon fixed by phytoplankton is incorporated into organic matter, which sinks when organisms die or defecate, carrying that carbon into the deep. The deep-sea organisms that consume this material incorporate some of it into their bodies, breathe some of it out as CO2, and in doing so participate in the global carbon cycle in ways that matter enormously for the long-term chemistry of the atmosphere. The deep ocean is not a sealed container. It breathes with the surface. It participates in the same cycle. But the vent communities, those discovered in 1977 and the many found since, are something different. They are islands of extreme productivity in an otherwise sparse environment, powered entirely by chemistry rather than light. The base of the food web is chemosynthetic bacteria, both free-living and symbiotic, that oxidize hydrogen sulfide, methane, or other reduced compounds to fix carbon. Above them in the food web come a variety of primary consumers, tube worms that harbor chemosynthetic symbionts in a specialized organ called the trophosome, mussels and clams, with chemosynthetic bacteria in their gills, a range of snails and crustaceans that graze on microbial mats. Above them come the secondary consumers, mobile crabs, fish, even occasional octopuses that arrive to exploit the vent ecosystem's unusual productivity. Vent communities are distinctive, and many of the organisms found at vents are not found anywhere else. They have evolved in isolation from the light-driven world above, adapting to temperatures that would cook most organisms, to chemical concentrations that would be toxic elsewhere, to the complete absence of seasons or day-night cycles. Some are known from multiple vent sites around the world, having dispersed along mid-ocean ridges. Some appear to be entirely local, isolated in a specific vent field, potentially for millions of years. The organisms associated with hydrothermal vents are windows into the ancient ocean. Not because they are themselves ancient. Most modern vent species are not particularly old, but because the environment they inhabit is ancient. The hydrothermal vent system, powered by the heat of the interior of the Earth, has operated since the Earth had an interior. The chemistry of serpentinization and magmatic heating, the mixing of vent fluid and seawater, the precipitation of minerals, all of this was happening 4 billion years ago as much as it happens today. The vent environment has persisted. What has changed is the biology that exploits it. Consider then the possibility that the lineage of organisms living at a modern vent field in the Pacific is connected by an unbroken chain of living things to the organisms that inhabited equivalent vent fields in the Archean Ocean 4 billion years ago. Not the same species, not the same genus or family or phylum, but the same fundamental ecological niche, exploited by the same basic metabolic approach in the same kind of environment. A thread of life so old it predates the continents, the atmosphere, the existence of any other kind of organism on Earth. Chemolithotrophic bacteria at deep-sea vents may be the closest living things to the original life, in terms of ecological context, if not in terms of exact phylogeny. Life at the edges, life in the dark, life in the most forbidding places the planet has to offer, not despite the difficulty but because of it, because difficulty means opportunity, in evolutionary terms, if nothing else is there to exploit it. The deep ocean was never empty. From the very beginning, something was down there, eating the heat of the Earth, building itself from the chemistry of rock and water, in the dark, in the cold, in the ancient and patient silence of the deep. There is a way to tell the history of life that focuses entirely on organisms, on the specific creatures that evolved, diversified, went extinct, left descendants. This is the way we usually tell it, but there is another way to tell it, and it produces a different kind of understanding. The other way focuses on processes, on cycles, on the ways in which the ocean and the atmosphere and the life within them are not separate things that interact, but a single system, a single interlocking machine of chemistry and biology and geology that has been running for 4 billion years. The carbon cycle is the most important component of this machine for understanding the history of life, and it is worth spending time on. Carbon is the backbone molecule of all life. Every organic compound that has ever been part of a living thing, every protein, every lipid, every nucleic acid, every sugar, is built around chains of carbon atoms. Carbon is fixed from the atmosphere and ocean into organic molecules by photosynthesis, and it is returned to the inorganic pool by respiration, by decomposition, and by geological processes. This cycling of carbon between living and non-living forms is not a background process that happens alongside life. It is, in a real sense, what life does. Life is the dynamic component of the carbon cycle. In the modern ocean, the carbon cycle operates through a combination of the biological pump and the carbonate pump. The biological pump moves organic carbon from the surface to the deep. The carbonate pump moves inorganic carbon in the form of calcium carbonate shells and skeletons built by organisms like foraminifera and coccolithophores, which sink when the organisms die and accumulate as carbonate sediment on the seafloor. Both pumps transfer carbon from the atmosphere, where it acts as a greenhouse gas, to the deep ocean and eventually to rock, chalky limestone and other sedimentary formations that lock carbon away from the atmosphere for millions to hundreds of millions of years. The Cretaceous chalk formations of England, the white cliffs of Dover, the gentle rolling hills of the South Downs are made almost entirely of the remains of microscopic organisms. Coccoliths, the calcium carbonate plates shed by coccolithophores, accumulated over millions of years in a warm, shallow sea. The chalk is biological product at a geological scale. The landscape was built by plankton. This is what the ocean has been doing for billions of years, taking carbon out of the atmosphere, incorporating it into living things, transferring it to the deep, locking it in rock. Without this process, the greenhouse effect of atmospheric CO2 would be far more extreme than it is. The ocean is the planet's long-term climate regulator. Life in the ocean is the mechanism by which that regulation operates. The oxygen cycle is equally fundamental. Photosynthesis produces oxygen, respiration and decomposition consume it. In a balanced system, these processes would nearly cancel out, but they don't quite cancel, because some fraction of organic matter, perhaps 1% or less, is buried before it can be oxidized and decomposed. Every carbon atom buried in an ancient shale or coal seam represents an oxygen molecule that was produced by photosynthesis and never consumed. Over geological time, this small imbalance has accumulated to produce the atmospheric oxygen we breathe. The oxygen in the air, in other words, is the accumulated surplus of billions of years of imperfect decomposition. The 21% oxygen atmosphere is not a stable feature of the planet in any absolute sense. It is the net result of a balance between burial of organic carbon and oxidative weathering of ancient buried carbon. When carbon-rich rocks are exposed by erosion and oxidized, oxygen is consumed. When organic carbon is buried, oxygen is released. The present balance, the atmospheric oxygen level that makes complex animal life possible, reflects the current equilibrium between these processes. Change the burial rate, or the weathering rate, and the oxygen level changes. Over geological time, it has varied considerably. The Carboniferous period, when enormous forests on the newly colonized land were burying vast quantities of organic carbon, saw atmospheric oxygen levels perhaps reaching 35%. The Permian-Triassic mass extinction, the most severe in Earth history, may have been accompanied by a crash in oxygen levels. The nitrogen cycle runs through the ocean in a different way, but with no less significance. Nitrogen is a component of proteins and nucleic acids. It is essential for life, but most organisms cannot use the molecular nitrogen, N2, that makes up 78% of the atmosphere, because the triple bond between nitrogen atoms is one of the strongest chemical bonds known and requires enormous energy to break. Only a small number of prokaryotes, the nitrogen fixers, possess the enzymatic machinery to do it. These organisms, by fixing atmospheric nitrogen into ammonia that other organisms can use, are the critical gateway by which nitrogen enters the biosphere. Without nitrogen fixation, life would be severely limited in its productivity. The entire biosphere depends on a relatively small number of microbial lineages with a very specialized metabolic capability. And those lineages are ancient. Nitrogen fixation is thought to have evolved early in the history of life, possibly in the Archean Ocean, possibly in a high-temperature environment, given that the enzyme responsible, nitrogenase, works best under conditions that suggest an origin in a warm anaerobic environment. The ocean was the site where this critical biogeochemical function was invented, and it has remained the site of much of the world's biological nitrogen fixation ever since. The nutrient cycles of phosphorus, of iron, of silica, of all the trace elements that life requires, run through the ocean too, each with its own dynamics, each linking geological sources to biological sinks, each linking the deep earth to the living surface. The ocean is not a container for life. The ocean is the circulatory system of the biosphere. The ocean moves things, atoms, molecules, energy, heat in ways that make life possible on the planet as a whole, including, eventually, on the land that the ocean slowly prepared. This is a concept that is easy to state and difficult to fully appreciate. The air you breathe was processed by the ocean. The nitrogen in your proteins was fixed by organisms living in the sea. The carbon in your bones was part of the ocean carbon cycle before it was part of you. The water in your cells has cycled through the ocean countless times. You are, in a very real chemical sense, made of ocean. You are made of the product of all those ancient cycles, all those billions of years of biological chemistry. The ocean is not outside you. The ocean is inside you, in the most literal possible sense. And so we arrive at the end of the story. That is not really an end at all. More of a doorstep, a threshold, the moment just before the next chapter begins, and you can feel everything that has been building, everything that the ocean has been quietly constructing across 4 billion years, poised on the edge of becoming something new. By the end of the Ordovician period, roughly 440 million years ago, the ocean had been supporting complex life for 100 million years since the Cambrian Explosion. The Ordovician ocean was spectacular in its diversity, an ocean full of trilobites, graptolites, cephalopods, brachiopods, echinoderms, early fish, reefs of sponges and corals in the warm, shallow epicontinental seas, a rich planktonic ecosystem of algae and small animals in the upper water column, a complex deep-sea fauna adapting to the conditions below. By almost every measure, the Ordovician Ocean was more biologically rich than anything that had preceded it in Earth's history. And then, at the end of the Ordovician, came the second mass extinction in the history of complex animal life. Glaciation, triggered perhaps by the removal of CO2 from the atmosphere by the newly evolving terrestrial plant life beginning to colonize the land surface, dropped sea levels, changed ocean chemistry, and wiped out perhaps 85% of marine species. It was not the largest extinction in Earth history. The Permian-Triassic extinction, 252 million years ago, holds that grim record. But it was enormous, and it reset the oceanic biosphere almost from scratch. The ocean recovered. It always has. In the aftermath of every mass extinction, the surviving lineages diversified into the vacant ecologies, new species evolved, new ecosystems assembled. The recoveries take millions of years. They are never precise replays of what came before. Each post-extinction ocean is different from the one that preceded it. But life returns. The ocean restores itself. This resilience, if we can use that word without making it seem like a plan, is perhaps the deepest fact about the ocean's role in the history of life. Not merely that the ocean was the place where life began and diversified, but that the ocean has an extraordinary capacity to absorb disruption and regenerate. Its sheer volume, the thermal inertia of all that water, the depth and complexity of its biogeochemical cycles, the diversity of the metabolic strategies employed by the organisms within it. All of these give the ocean a kind of systemic durability that no terrestrial ecosystem can match. Mass extinctions, glaciations, asteroid impacts, volcanic winter, the ocean weathers them all. It takes longer to die than anything else on Earth, and longer still to recover. But it recovers. The colonization of land, when it finally happened, was not the ocean releasing its children to a better life. It was organisms adapting to a new environment at the ocean's margins, driven by the same pressures that have always driven evolution: competition, resource availability, the constant search for an unexploited niche. The first land plants, appearing in the Ordovician-Silurian transition around 450 to 430 million years ago, We're essentially aquatic algae solving the problem of desiccation. The first land animals, arthropods, almost certainly, perhaps millipede-like creatures or early arachnids, followed the plants or accompanied them, exploiting the growing terrestrial resource base that plant life was creating. The fish that eventually made it onto land in the Devonian were doing something no vertebrate had ever done. But they were doing it with bodies built by 400 million years of marine evolution, with lungs that had evolved in water, with limbs that had evolved as fins. Nothing about the terrestrial biosphere was invented from scratch on land. Everything was adapted from marine origins. The respiratory systems of air-breathing animals are modified versions of organs that originally worked in water. The limbs of tetrapods, the four-limbed vertebrates that include all amphibians, reptiles, birds, and mammals, evolved from the paired fins of fish. The sensory systems, the cardiovascular systems, the immune systems, all of it traces back to the ocean. Land life is ocean life that moved. The ocean is the template. Land is the copy, edited for a different medium. This is not a small thing. When you look at a tree, you are looking at a modified alga. When you look at a bird, you are looking at a fish that kept diversifying for 400 million years and ended up with feathers. When you look at your own hand, you are looking at the rearrangement of bones that first appeared in the fin of a Devonian fish 400 million years ago in a river delta somewhere in what is now the northern hemisphere. The continuity is not metaphorical. It is biochemical, anatomical, and phylogenetic. The ocean is inside the tree and inside the bird and inside the hand. We look at the ocean now and see something outside ourselves. A resource, a destination, a danger, a source of beauty. We stand at its edge and feel something, awe or longing, or a mild unease that has no easy name, and we have theories about why. Some evolutionary psychologists suggest that humans feel drawn to water because our ancestors lived near it, because proximity to water meant survival. Some poets say we feel drawn to it because the ocean is what we came from, and the body remembers what the mind has forgotten. Maybe both things are true and maybe neither is exactly right, and maybe the feeling is just the feeling. The human encounter with something old beyond comprehension, indifferent to our presence, carrying on as it has always carried on, whether we are here to notice it or not. 4 billion years. That is the number we keep returning to. The ocean has been doing this for 4 billion years. Life has been living in it for most of that. The cycles have been running. The chemistry has been cycling. The organisms have been dying and being replaced by descendants slightly different from themselves. Generation by generation, extinction by extinction, recovery by recovery, for 4 billion years. Our entire existence as a species, Homo sapiens, present on this planet for perhaps 300,000 years, literate for 5,000, industrial for 200, is less than a rounding error in that timeline. The ocean was ancient when the first fish climbed onto land. It was ancient when the first dinosaur walked. It was ancient when our primate ancestors descended from the trees. It has been ancient for longer than any word for ancient can contain. And yet it is not indifferent to us in the way that, say, a rock is indifferent to us. The ocean is a living system. It responds to what is put into it. It has been responding to human activity for decades now. Warming, acidifying, losing oxygen in its deeper layers, accumulating plastics and chemicals, and the thermal excess of an industrial civilization that burns fossil carbon. Those fossils are, themselves, ancient marine life. The compressed bodies of plankton that lived and died in Mesozoic oceans, stored in rock for hundreds of millions of years, and now being released in a geologically instantaneous burst. We are, in a real sense, burning the ancient ocean, taking what the ocean carefully locked away and releasing it back into the air, reversing in centuries what took millions of years to accumulate. The ocean will absorb this. It will absorb it the way it has absorbed every disruption in 4 billion years of history. Slowly, imperfectly, at the cost of many of the organisms currently living within it, and with consequences that will play out over thousands of years. The ocean is not fragile in the deepest geological sense. It has survived impacts, glaciations, mass extinctions, changes in atmospheric composition far more extreme than anything we have yet produced. But it is not fast, and we are fast. The gap between our speed and its speed is where the damage happens. This is not the place to make an argument or deliver a verdict. The facts speak for themselves, and the listener will form whatever view they form from them. But there is something worth holding some consideration that feels important before we close. We are the first species in 4 billion years of life on this planet that has ever had the capacity to understand what the ocean is and what it has done. Every organism that came before us participated in the ocean without any possibility of knowing it was participating. The bacteria at the hydrothermal vent has no idea it is part of a biogeochemical cycle that regulates the atmosphere of a planet. The trilobite in the Ordovician Sea had no capacity to know that it was part of the richest marine ecosystem that had existed in 300 million years. The Ediacaran organism, whatever it was, had no way of understanding that it was one of the first complex multicellular life forms to ever exist, that it was sitting at a hinge point in the history of life. They simply lived. They simply did what the chemistry of their bodies required them to do. We are different. We can know this. We can understand the depth of the ocean's history, trace the lineages, read the isotopes, reconstruct the ancient environments, tell the story. We can hold in our minds, however imperfectly, the 4-billion-year arc of life in the sea. We can feel the weight of it. We can feel the debt. This capacity for knowing, for understanding the context in which we exist, the history from which we emerge, is itself the product of that same 4-billion-year story. The brain that allows you to understand the ocean was built by evolution operating in organisms that came from the ocean, and still carries, in its basic architecture and chemistry, the signatures of that origin. The neurons in your brain communicate using ions, sodium, potassium, calcium, whose concentrations are regulated to maintain something very close to the ionic composition of ancient seawater. The composition of your blood plasma, its saltiness, is not arbitrary. It is a memory, an echo. The chemical legacy of the sea in the fluid that carries oxygen to your cells. We are the ocean, thinking about itself. We are what 4 billion years of chemical evolution in the sea eventually produced, life complicated enough to turn around and wonder about its own origins. That seems somehow the most remarkable thing of all. Let me tell you about a place. It is not a place you can visit because it no longer exists. It existed approximately 3-5 billion years ago, in a part of the world that is now the Pilbara Craton of Western Australia, though the geography of that time would have been unrecognizable. The sea was shallow there, warmer than any modern sea, moving under a sky without oxygen, lit by a sun that was dimmer than the one above you now, but still powerful enough to drive chemistry on the water's surface. The bottom was soft sediment, and on that sediment, thin mats of microorganisms lay in laminated sheets, growing slowly toward the light. The water above them was silent in the way that water at depth is always silent, not the absence of sound, but the particular silence of water where sound moves differently, where the vibrations of the world above are filtered and muffled and arrive changed. Nothing in that shallow Archean sea was looking at anything else. Nothing had eyes, nothing had nervous systems, or brains, or the capacity for awareness. The organisms in those mats were not aware of the light they were growing toward, were not aware of the sulfur compounds they were metabolizing, were not aware of each other in any meaningful sense. And yet they were alive, and yet they were doing something, growing, dividing, extracting energy, building those layered structures that would one day be found by a species they could not have imagined, in a world that would not exist for billions of years. There is a kind of beauty in this that resists easy articulation. The beauty of deep time. The beauty of process without consciousness of becoming, without intention of life unfolding in utter silence and darkness and slowness, and producing in the end, something that can stand at the edge of the sea and feel moved by the sound of the waves. There is something about knowing this, knowing the full depth of the story that changes how the ocean feels. Not just water, not just geography, something older than any category we have for old things. Something that was here before the mountains, before the air, before the stone beneath your feet. When you look at the ocean, you are looking at the environment in which life began. Whatever you believe about the world, whatever you carry in your philosophy, or your faith, or your uncertainty, that fact is there. The ocean holds its beginning still, somewhere in its water, in the deep sea vents that still vent hydrogen and mineral-rich fluid into the cold dark, in the chemistry of its ions and its salts. The conditions that made life possible are still there, not identical. The ocean has changed enormously in 4 billion years, but related, continuous. Descended from those original conditions the way you are descended from those original organisms. The ocean remembers in the only way that chemistry and physics can remember, by continuing, by keeping the cycles running, by cycling carbon, fixing nitrogen, precipitating minerals, maintaining the conditions, however changed, however stressed, under which life persists. I want to say something here that I'm not sure I can say correctly, but I'll try anyway. There is a kind of comfort in the deep time of the ocean, if you let yourself feel it. Not the comfort of being told that things will be fine. The ocean's history includes far too many extinctions and catastrophes for that kind of comfort, but a different kind. The comfort of scale. The understanding that what we are and what we are doing, and even what we fear, all of it is happening inside a process that is so much larger and older and more durable than any of it. The ocean was here before us. It will be here after us, in some form. Life was here before us and will be here after us in some form in all likelihood. Not our form, not our lineage, but life, the chemistry that continues. That is not a reason for complacency about what we are doing to the ocean. Nothing about the largeness of geological time makes what happens to the living ocean in the next 100 years irrelevant. Those things matter enormously to the organisms alive today, to the people who depend on them, to the ecosystems that have taken millions of years to assemble. Extinction is permanent. A species lost is gone from the story forever, even if the story continues. But there is still something stabilizing about the view from deep time, about standing on the shore and knowing, really knowing what the water in front of you is. Not just ocean, the stage on which 4 billion years of life was written, the medium in which the first chemistry crossed into something more than chemistry, the system that produced every organism that has ever lived, including you, including everyone you have ever loved, including every creature you have ever looked at with wonder or affection or fear. Have you ever stood at the edge of the sea at night, when it's dark and the water is black and the sound of the waves is the only thing, and felt, without quite knowing why, that something very large was present? Something too big to fit into any single thought, but pressing against the edges of your awareness, like a question that doesn't have words yet? Maybe that feeling is simply the ocean's size, and the darkness, and the sound. Maybe it's the evolutionary inheritance of an animal that needs water and knows, somewhere below language, that it is not separate from the water. Maybe it's something else entirely, something we don't have the framework to name, but maybe it is also, in some small part, the actual depth of the thing, the actual weight of what is in front of you. 4 billion years of history contained in that water. 4 billion years of chemistry and evolution and catastrophe and persistence. The beginning of everything you are, and everything you know, and every form of life that has ever existed on this planet. All of it present in the water. All of it continuing. There is a moment sometimes when you can feel it, when the scale of it arrives briefly and undeniably, and the familiar world, the road behind you, the phone in your pocket, the ordinary concerns of ordinary life, all of it becomes very small for a moment, and what is left is just this: the water and the dark and the extraordinary fact of being here at all. The extraordinary fact of being alive, in an ocean world on a planet that took 4 billion years to produce something capable of knowing it was alive. The early bacteria knew nothing. They knew nothing at all. They grew toward the light and divided and changed, and eventually became everything. They had no idea. They had no idea what they were starting. And somehow, improbably, incomprehensibly, it led here, to the shore, to the dark, to the person standing at the edge of the water in the night, looking out, not quite sure what they're feeling, but feeling it anyway. That old, deep, pre-verbal pull toward the ocean. That recognition, maybe, of what it is, what it has always been. The water was there first. It was there first, and it is still here, and everything you are came out of it.